have been mainly working in the last 10 years with natural aquatic systems. But back when I did my PhD, I worked with wastewater. And now again, as you mentioned, I work again with wastewater, although from a very different perspective. And I have been also teaching microbiology. And I always considered wastewater like a great example of how we can harness the power of microbes for our own good. So that's why what I will present here in the first half of my presentation is not so much my research, but it's more like wastewater microbiology in general, and not always it will be more of a lecture, an educational lecture. And at the end, I will present a little bit of what I have been doing in the last 18 months. So wastewater microbiology. You are at the end of a course where you learned a lot about microbial ecology. And usually we focus on how they interact with nature and how fantastic they are as biological entities and ecosystem drivers and so on. But microbes also provide us a lot of services. So, and a lot of these services are connected to their function in the biogeochemical cycles. I don't know if you know this paper, Life in the World Without Microbes. It's a thought experiment about how would be the life on Earth if we don't have microbes. It's a short and easy to read paper. I can recommend it. But if you want an even shorter version, then there is a YouTube channel by Ed Yong, and he has a set of awesome educational videos about microbes, and one of them processes this is about this study. But basically, what the authors, Gilbert and Neufeld, said in this little paper is that without microbes, of course, we wouldn't have diseases and decay, and that's good. But otherwise, we just would lose a lot of the good thing in life. We wouldn't have beer or wine. We would have digestion problems, no vitamins. Our crops would fail. Our livestock would die. No one would degrade. We will pile up dead corpses all around us, and also the oxygen production will be halted, and after time we would use our oxygen. So, but I think I don't have to prove this too much. Microbes are really useful also for us as humans and provide us a lot of ecosystem services. Today here I will focus on wastewater and how wastewater treatment connects to the carbon, nitrogen, and phosphorus cycles or biochemical processing of microbes. Why do we need wastewater treatment? Well, if we don't treat our wastewater and just let it out to the natural recipient aquatic systems, then we will let in a lot of nutrients, and nutrients cause eutrophication. I suppose you are familiar to some extent with eutrophication. This is one of my favorite figures demonstrating the power of, for example, phosphorus in eutrophication. Is an experiment from the 80s when they divided the lake into two, and they added phosphorus to one side, and it turned as green as this. I won't go into the details of eutrophication, but basically, for example, if wastewater goes into a river, then all the nutrients and carbon sources added will be used by the microbes. There will be a great reduction of oxygen, a big growth of bacteria. Then later on, all the nutrients will be used by algae. There will be the blooming, this greening process, and the decay of that will further reduce the oxygen, and it will be quite long down the river to recover of such input. So this problem with letting our waste into the nature has been recognized long ago, as in this little, what is it, how you call biblical phrase. So This one, there are so many versions of English Bible, I realized when I was looking for this one. So I just took one that's not too direct. So carrying a paddle, a tie girdle, and when thou sits down, thou shalt dig round about, and with earth that is dug up, thou shalt cover. Maybe I don't do it in good English, but you get cover what your excrement. Nowadays, we have more sophisticated systems to do this, to protect our recipient aquatic system. This is a wastewater treatment plant. I'm sure you have seen this kind of systems here and then with these circles and big tanks and bubbling water and so on. So today I will guide you a little bit of what happens in a system like this and how microbes contribute to what's happening. This figure is from the biology book of my daughter in 7th grade. And I put it in because sometimes students tell me, why do we need to know how a wastewater treatment plant works? So if it's even in the 7th grade biology book, maybe we can know a little bit about it. And also I think because we are all contributing to the ways that it's treated here. So it's good to know what actually happens. We can have also actually a more complicated description of what happens. And it's important to know that there are many, many different settings for wastewater treatment systems. So what I want you just to know is that usually we have a primary treatment first, which is a mechanic treatment where the big things are removed. A lot of trash and oil is removed with basically just mechanically with big sieves and so on. And a lot of the solids is already removed here and moved to the sludge processing that I will talk later. Then comes the next one, the biological treatment or secondary treatment, which is a mostly aerobic treatment and where a lot of the carbon removal happens already here, but also nitrogen and phosphorus gets removed. And then in many places, we only have these two levels, but depending on how sensitive is the recipient water, we usually also have a tertiary or third treatment level, where further removal of nutrients, nitrogen and phosphorus happens either biologically or chemically or with both. And sometimes there is even a disinfection to avoid the release of pathogens to the recipient water. And as I said, there is also a sludge processing line that doesn't flow out to the recipient. So now let's start first how the carbon cycle contributes to these processes. This is from a Swedish textbook, so it's in Swedish, sorry for that, but I think it's quite easy to get the words, and we don't want to go now through the carbon cycle in total. What matters is that the carbon cycle has both carbon sink processes, when autotrophs convert CO2 to organic carbon, and also carbon source processes. Now the sink and source is related to the atmosphere. And in the source processes, we have mainly when heterotrophs convert organic carbon into CO2, and when methanogens, when through methanogenesis, CO2 is converted to methane. In wastewater, we don't really focus on the carbon sink part. Wastewater treatment is primarily a carbon source process. But we have different aims. So basically, we have a lot of organic carbon in the wastewater. We want to convert as much of it into inorganic carbon. So both of these processes are good for that. And a further goal in wastewater treatment is to do as much as possible through methanogenesis. So we want to produce more methane than CO2. Why? You would think that CO2 is lesser greenhouse gas. We want to get more dust and less methane, but no, because in wastewater treatment, this process happens in a closed controlled system, and the methane can be collected and used as biogas, as a fuel. So that's why the aim is to get as much of the carbon into the anaerobic line. So briefly about the primary wastewater treatment plant. As I said, this is a physical removal of solids. So these are the typical settler, they're called clarifiers. Sorry, so they call these ones clarifiers that you can see if you pass by a wastewater treatment plant. In these clarifiers, if you just look at this, you don't see anything. If you stop there and look them for 5 minutes, then you will see that these things here go around really, really slowly. Like here, we can see it goes around really slowly. And so it doesn't want to mix up the solution, but it helps the sedimenting solids to move them around and move them to the center and be able to remove them. And from here, they will go to this sludge treatment. Okay, should I, do we check the chat later, right? That's what we said. Okay, okay. I just noticed now. And so that's the sludge part, but the liquid goes further to the secondary treatment. So now the secondary treatment is already a biological treatment. And what happens here is that we have first an aerated tank, where by aerated means that pressurized air is bubbled into the system. This is if you've got a wastewater system, you see these bubbling liquids, very liked by TV crews to show it. And what happens, the goal of the aeration is to give oxygen for aerobic heterotrophic processes. Why is it called activated sludge? Well, it's called activated sludge because it's not just water, it's a water full of flocks, or in limnology, we would call them particles, I think. So these flocks are basically mini biofilms, are like small islands of microbial growth. And what happens in these flocks is that the same way as, for example, in an ocean or a lake, we will have much more biological activity connected to particles than in a free living state. Basically, the aim is the same in the wastewater, that we have these flocks that have a lot of bacteria close together, and the degradation of carbon compounds happens much faster than if it would be just free. And this is why it's called sludge, and now you get the name, activated sludge. And to form this kind of flocks, there is one species that is very well known that usually gets very abundant in this system, and that's the Zooglea ramigera, which is a gram-negative aerobic flock-forming bacteria, meaning that it produces all these extracellular polysaccharides that can form these sticky chunks. And if you would go here, take a bucket of water and let it settle, then it will settle like here. Actually, this is a very important factor of the activated sludge that's tested on a regular basis at wastewater treatment plants, how well it settles. Because after this bubbling part comes again a clarifier, the same, this round thing going around, and the sludge part will be settled and flown to the sludge line. So basically, by building these flocks, the bacteria collects in its biomass all the carbon and a lot of the nitrogen and phosphorus, and then we can settle it down and remove it from the water. So, so that was the aerobic part of the carbon cycle, but we also use, as I mentioned, we want to get more methane, and for that we need the anaerobic carbon degradation. Anaerobic carbon degradation, I don't know how much you talked about methanogenesis, but basically it's the it's working together of archaea and bacteria. And it goes from complex, it happens completely without oxygen. So if we have oxygen, then this process won't work. And basically complex polymers get first degraded to monomers. Then we have fermentative bacteria that will convert this to CO2, but also to different small organic acids. And then depending on the ratio of these two, and hydrogen, sorry, and depending on the ratio of hydrogen and organic acids, and also depending on the type of methanogens that are present, we will either have the acetoclastic methanogenesis, or we will have the hydrogenotrophic methanogenesis. And as you can see, there is more methane. There is only methane here, while here is more of a mix of CO2 and methane. So there is a lot of research to tweak these systems to go more into this way, the only methane, because then the biogas produced will be more pure and more efficient to use as a fuel. This is a complete, gigantic, big research field of how to tweak fermenters to go on what way, on one of the ways, and also on how to tweak fermenters to process all kinds of things, for example, antibiotics. But I won't go into details. I think if you're interested, you can really read a lot about this. But I will talk a little bit of what happens in these sludge treatment reactors. So there is no oxygen, all this sludge comes in. And again, there is some sort of upwelling, bubbling sort of thing. But here the bubbling doesn't happen because air is pumped in, but the bubbling comes from all the methane that's being formed, which is harvested and used. And the methanogenesis, again, is not happening mainly by free-living bacteria, but happens on so-called sludge granules. And these are really amazing things. I will go to my next slide. So a sludge granule is like a mini consortia of all of these bacteria and archaea that do this whole anaerobic carbon degradation process. And if they do some fish staining with archaea, and here the red ones are archaea specific probes and the green ones are bacteria specific probes, then we can see a clear layering of the two. So this is a transsectional cut, and this is from outside. And in the middle will be the methanogens that need this the lowest oxygen and the lowest electron potential. And the outside layers are the fermentative bacteria. So it's a mutualistic organisms. And so the mutualism has even a special level when it comes to anaerobic carbon degradation and these granules, and that is syntrophy. And syntrophy, I don't want to go too much to the details because I always get lost with this, but the point is that syntrophy is an energetic mutualism, which allows organisms to survive with metabolic pathways that don't produce energy, but instead require energy. So one of these is ethanol fermentation that, as you can see here in the delta energy, needs energy, but produces a lot of reductive power. And then by providing this reductive power to other organisms, in exchange, it gets, so here is how it provides this hydrogen transfer to the methanogens. In exchange, it can it gets a transfer of it can survive energetically. Again, this is I'm not an expert on this, and there are a lot of studies, for example, looking into how this kind of energy transfer can happen between bacteria and archaea, and it's a super exciting field that for those interested, you can read a lot about it. And so one more about these granules. This is from a study done here in Stockholm into pilot, so experimental little reactors. And here they looked into how different sized granules. So these granules you can see by eye even. They are small but visible, have different structures, and that's one of them is multi-layered. These bigger ones, the smaller ones don't have so many layers, and then depending structure they have different methanogens. So one is more of this acetoclastic that produces the mix of methane and CO2, and the other one is more of the hydrogenotrophic. Again, by tweaking the reactor performance, you can switch them from one to the other. Okay, time is running, so let's move to nitrogen. So the nitrogen cycle is my favorite of all cycles. It has a lot of different reactions, both aerobic and anaerobic. And here we also have the nitrogen gas as the starting point. And we want to look into how this is transformed into organic nitrogen in wastewater. So I will remove that, not to confuse us. It's not interesting in this case. Our aim in wastewater treatment is to go from our little organic nitrogen provider into this inert and harmless nitrogen gas. And for this, we will need, first we get ammonia. Then this ammonia will be first converted into nitrate, and then under anaerobic conditions, the nitrate will be denitrified into nitrogen gas and released. And then there are some variations for this process. We have since 2015, we know that there are organisms that can do this in one step, the Comamox organisms. And we also know that there is an anaerobic process that directly uses ammonia and nitrate to produce nitrogen gas. So that's an alternative denitrifying line called anamox. And all of this can be used in wastewater. I will show you. So some of this nitrification, the nitrification happens already in the activated sludge step, but mostly when they are preferentially enhanced, that happens in the tertiary treatment. And in these treatments, usually the process, as I showed you with the cycle, has first an aerobic step where we enhance nitrification. So going from ammonia to nitrite, and this needs a lot of air to be pumped. Pumping in air is money. This is important to note. And then comes the denitrification step, when we go from nitrate to nitrogen. And this one is actually not autotrophic, but a heterotrophic process and needs organic carbon source. So usually they add a lot of methanol here to enhance this process. And that's, again, a lot of money. So there are some ideas to make this cheaper and using microbes. So one of them is to tweak the system a bit towards this anamox process that I mentioned, where ammonia and nitrite are used in an anaerobic way to produce nitrogen gas and water. The thermodynamic possibility, this is an amazing process, of this process has been already described in 1977, but no one knew if this really exists. So basically, Broda just said that if we look at the energetic balance, this reaction should exist in microbes, but no one knew if it really exists until 1999, when specifically aiming for this process that should exist, anamox organisms were discovered, and it's a Planktomycetes group. And so these are anaerobic chemolithoautotrophs, meaning that they don't need carbon sources. So, and they don't need air. So basically we can save on not aerating and not needing to add methanol or other carbon source here. Just we have to tweak the system. The problem is, if you remember, this needs nitrate and not nitrite, nitrite and not nitrate. So how to tweak the system? So here comes a solution. So instead of having a nitrate here, we need to have a mix of ammonia and nitrite. So as I mentioned, nitrification happens in 2 steps. So the first step is ammonia oxidizers going from ammonia to nitrite. These are faster growing. And the other one is the nitrate oxidation when we go from nitrate, and these are slower growing. And if the temperature is higher. So one possibility is to increase here a bit the temperature, and then this process won't have enough time to happen properly. So we will end up with nitrite instead of nitrate. And basically, just by tweaking this, the system can be changed to go into an anaerobic system. And that saves a lot of money. So now I'm done with nitrogen. Briefly about phosphorus. So phosphorus removal a lot of times happen chemically. This is important to know, but there are solutions to do it also biologically, and I will only talk about that. So the biological solution for phosphorus removal is called enhanced biological phosphorus removal, and happens with help of very cool bacteria. And here, this happens at the activated sludge level, so the secondary treatment, by adding an anaerobic trunk before the bubbling aerobic trunk. And the aim of these changes is to promote the growth of polyphosphate accumulating organisms, or PAOs. And these PAOs will incorporate phosphorus into their biomass, and then in the clarifier, they can be removed and sent to the sludge line where they are later processed. So what do PAOs do? So the most well-known is, again, a Candidatus species, Accumulibacter phosphatis. And what this one do is in anaerobic conditions, it uses volatile fatty acids and produces a sort of these ones using anaerobic metabolic pathways, produces polyhydroxyalkalonates, which it accumulates. Like if you do a microscopic picture of this bacteria, then you can see like this bubble kind of, they are also called bioplastics. So basically it like accumulates sort of bioplastic pearls inside of its cell. And to do this accumulation, it uses ATP from accumulated polyphosphate, and also releases, but when hydrolyzes the ATP, releases phosphor to the system. So this is not good. It's releasing phosphor when we want to use it to remove the phosphor. So then comes the aerobic phase, when actually it will utilize all these collected PHAs, so these little plastic pearls, and use this as energy source. And for that, when it hydrolyzes it, it will produce the polyphosphates. And so basically, just to keep it simple, here is also a graph. Under anaerobic conditions, it uses volatile fatty acids, releases phosphorus to the water phase, and inside the cells accumulates these polyalkalonates, while in the aerobic phase, it will use these polyalkalonates, polyhydroxyalkalonates, and will accumulate polyphosphates. And basically accumulates more polyphosphates in the aerobic phase than what it releases in the anaerobic. And if I could go back to my previous slides, so after the aerobic, we have the clarifier. So all this phosphorus is in the biomass and can be removed and sent to the sludge line, and that's the phosphorus removal. Okay, so now I used almost all my time, but I got to the end of what I wanted to say about wastewater treatment. I will very quickly talk about what I do now, because as Yasmin mentioned, I have been working with non-wastewater, but then life turned me back to wastewater, but from a very, very different angle. So actually my plan for summer 2022 was to go to Siberia and sample some very cool permafrost lakes and do some virus and bacteria studies and so on. But yeah, nothing happened of this. And then I had to look for something else for that summer, at least. That's what I thought. So there was a call, who wants to do something related to COVID-19. And then a few of us thought, oh, maybe then I don't, instead of like working on all that old stuff I have, maybe I could do some new thing. And the idea came that we could look for COVID-19 in wastewater, or the virus causing COVID-19. So I will jump over here. So why can we do this? Well, basically very early on, already in March, maybe already February, there were some reports that in 2020 that the COVID-19 virus, SARS-CoV-2, can not only be detected on the nasopharyngeal swabs, but also in fecal samples and in quite large amounts. And then in March, the first report of coronavirus being detected and in Dutch wastewater came along. So this was our little project that, okay, we will look into wastewater. We will look for SARS-CoV-2 wastewater, for SARS-CoV-2 virus in wastewater to see how many people are infected. Immediately, there was also, of course, a concern that maybe this is also a potential infectious source. But SARS-CoV-2 is an envelope virus, and usually envelope virus, even if they can detect it in feces, by the time they go through our GI tract, they are not infectious. And luckily, this seems to stand to this day that the transmission through the fecal route is almost non-existent for SARS-CoV-2. So this also made our work easier, because then we don't really need some BSL 3-4 facilities to do this project. So, but what is the concept of the project? The concept is that when it comes to wastewater, we all contribute to it, all of us who are connected to the system. And we do this usually daily, and we do it anonymously. So our contribution doesn't need any, wastewater analysis don't really need ethical approvals or similar. And also we all do it. So if we are asymptomatic, we still will contribute to the wastewater. If we don't want to get tested, we still will contribute to the wastewater, and so on. So wastewater can tell us, in this sense, a less biased view of the current prevalence of an infection. And we can quantify the amount of virus in the wastewater and give a proxy for this prevalence. And right now we are also working on sequencing the virus from wastewater to look, for example, for the ratio of Omicron variants right now. And then this kind of information can help guide public health. So, but this is not as simple as one thinks, because to start with, wastewater is not only what comes from our toilet, but of course it comes also from other parts of our bathroom. But many systems also get input from rainwater, even some smaller streams can add to wastewater systems, and they can be also industrial water. So the water flow comes from different sources, and only part of it is what we are interested in. Then, so we decided, therefore, we need a good control that's already in the wastewater. And very early on, our choice fall on a pepper mite multivirus, which is interestingly, this is a plant virus, a virus that infects chili and paprika and other similar plants, causes them to look like this. And interestingly, this is the most abundant RNA virus in human feces. And it's not present in animal feces. Animals usually don't eat chili. So it has been used for a decade as a human fecal pollution indicator. And yeah, so what we use this virus for, but we can also quantify it from the wastewater and use it like a proxy to quantify how many people are contributing to the wastewater. Our other big target is SARS-CoV-2. This is a really nice model of it. So SARS-CoV-2, as I mentioned, it's compared to this pepper virus. This is a non-enveloped virus. This is an enveloped virus. And it's an RNA virus. Both this and the pepper virus are RNA viruses. That's also a good help for us because we can use the same molecular processes to analyze them. And what else? Yeah, I think you know very well this virus. I will jump over. So a bit how the sampling happens. So wastewater can be sampled in many ways. One is just to grab a sample from a wastewater system. Another is to use continuous samplers, so that sample all the time. But then the problem comes that the wastewater flow varies. So in the morning, people use a lot the bathroom, like here, then less, then in the night, they barely use, then in the evening, they use it again, and so on. So, but luckily, every little bit more modern wastewater treatment plant is equipped with so-called flow compensated auto samplers. So these samples take water depending on how much water is flowing through. And this is what we use. So basically, every week, one day, we take a sample, we get a sample like this from the wastewater treatment plant, and that will be very representative of that day. Then we take this sample to the lab, and we have to concentrate first the virus because it's just a very little part of all the crap that's in the wastewater. Then we extract the RNA, and then we detect it and quantify it using quantitative PCR. First reverse transcription and then quantitative PCR. And this is a slide a little bit old now. So this is some of our monitoring. So we quantify both the SARS-CoV-2 virus and this pepper virus, and then we look for the ratio of the two, and we use this as a prevalence proxy. So here you can see some of our results. You can see that this summer it got really, really low, but luckily we have worked a lot on making the methods much more sensitive. So even when it was low, we could see different trends if I switch to a log scale, and we could like see smaller outbreaks and so on. With the help of these methods, so I work mainly here in Uppsala, but as you saw, we got also samples from other places. We were able to predict a little bit the second and the third wave. And unfortunately, right now we are again seeing an increase. Let's hope it's not one more big wave like we see here. And yes, so it relates relatively well both with the cases and hospitalization. And so as I said, it's quite good to see local outbreaks. So this is again our data. You can see this green one. This is what I will be talking of now. So first we thought maybe something's wrong with our measurement, how it got really that high. This is Karlebu. It's a smaller municipality north from Uppsala. And you can see it was always a bit higher than elsewhere, but this was unusual that we had this peak in October. And then we looked at the cases, and actually it was the highest case rate by 100,000 people and highest positivity in the same season when we had this peak, and also the cases showed a peak. So basically it showed that this method is quite good to detect these outbreaks. So if once maybe we move to life where we don't test every cold because like we have so good treatments, or I don't know why, then wastewater can still tell us if there is a lot of COVID-19 going around. And this had some correlations also to vaccination, but I think I jumped this over now. And yeah, so we can also use this method to look for neighborhoods. So this is a pilot study that we ran here in Uppsala to look for we chose some regions that are socioeconomically more disadvantaged and some that are exactly the opposite. And we did see that in the more socio and economically disadvantaged places, we had unfortunately more COVID than in general in the town or the richer regions. And yeah, so wastewater monitoring is supposed to be done by every EU member state already from this October. It's still not happening that much. We have also other projects. We are trying now to use this method in Uganda, where testing is really, really low. But we are also hoping to use wastewater not only for SARS-CoV-2, but to study a lot of other things. And this is a picture from a review before the pandemic showing for how many things wastewater can be used. So you have to know now, connecting to the beginning of my lecture, that this is all the wastewater before the treatment, because a lot of things get luckily removed by these amazing wastewater treatment plants. But if we look into the wastewater before treatment, the raw wastewater, then we can deduce a lot of useful information about the health of the population that's producing the wastewater. So we can look into what kind of drugs they use, both illegal and legal, what kind of pharmaceuticals they use, what kind of industrial contaminations they are exposed to, and even things like what kind of diet, there are diet markers that can be measured. And of course, what kind of pathogens they are being infected by. So that's all what I had, and sorry for being so long. Of course, I do this wastewater with the help a lot of people, and I won't name now here everyone. This is my small group here at Uppsala, and I also like to thank everyone who donates anonymously to us for this project. So thank you very much.